So like I said, nucleic acids kind of important for what's going on right now. And the other thing that I sort of thought was interesting about it is that since we just finished talking about sugars, you will see that carbohydrates are extremely important in nucleic acids because they're based on a sugar called ribose. And it's a 5-carbon sugar, and it's a part of every DNA and RNA nucleotide. So knowing how sugars can, the different reactions that sugars can undergo, and having learned that hydroxyl groups are really, really important in sugars as being a target for attack, as being a target for oxidation, we can hope gives us a little more understanding of the difference between DNA and RNA and why one of them became basically was selected as the repository of all our genetic material and the other one wasn't. So let's start. So we want to identify different types of molecules that combine to form nucleotides. We're going to see that they're formed of phosphates and phosphate diesters. And we certainly know something about esters, but we haven't seen phosphate esters before. So that's a new type of bond we're going to be looking at. Sugars and then the bases that go on top of the sugar. We're going to learn about RNA and DNA, and then how nucleotides are linked together to form long nucleic acids. And it's the sequence of these nucleic acids that is the repository of genetic information. So the specific sequence that they lay down is what codes for proteins and RNA and all the other things that your body makes. So we're going to talk about the structure of DNA. You probably are familiar with the old double helix. I'll tell you a little bit about the history of that because it's really, really interesting. And how the structure that discovered double helical structure explained, once that was made apparent, it sort of, tore the lid off of all of biology and biological chemistry, because it explained so beautifully how DNA replication works, because that was something before that nobody really understood how it worked. It had been known that DNA was the repository of information for a really long time, but nobody understood how it worked. They knew that it was made out of bases, they knew it was made out of nucleotides, they knew that there was a sequence, but there's no way to sequence it, and there was no way of knowing what the structure was. And without the structure, there's really no way of understanding it. So we'll go over that and show how the double helical structure explains perfectly how DNA is replicated. And then we'll talk about how RNA is made from DNA and the really, really important differences between RNA and DNA. And that is one of the main things I sort of wanted to hit on, because COVID-19 is a virus where its genetic material is actually RNA rather than DNA. And we'll see why that's really important. So we'll talk about different types of RNA. You may be familiar with messenger RNA. That's the RNA that's translated into proteins. But there are 2 other types of RNA that are, I don't know if they're just as important, but they're pretty damn important. And then we'll talk about the genetic code. You're probably familiar with the fact that proteins are encoded in our DNA in terms of 3 nucleotide codes. And that code was broken down in the late 50s, early 60s. We now sort of understand how the sequence of a DNA, we can then take that sequence and we can automatically know what the sequence of that protein is going to be. We may not know exactly what shape it's going to take, although we're getting better at that with supercomputers and everything else, but we can certainly figure out what the sequence is. Then we're going to talk a little bit about genetic disease. And and the different types of mutations that can take place in DNA. And lastly, I wanted to hit about how viruses reproduce in cells and why it's sort of the different ways that DNA viruses and RNA viruses replicate themselves explains a lot of what's happening as far as COVID-19 is concerned. All right, so first let's talk about chromosomes and genes. So chromosomes, you've probably seen pictures of chromosomes. There's a picture of a chromosome right there. And the way that it's structured is basically you take that very simple double helix and you just loop it on itself and make another helix out of it. And you keep twisting and twisting and twisting and twisting to get this sort of super helical structure with proteins in it to keep it all compact. I know that none of you have like phone cords anymore. When's the last time anyone had a phone with a cord on it? But maybe if you went to your grandmother's house or your great grandmother's house or whatever, you've seen those old phone cords that had a swirl in them. Well, if you just kept turning them and turning them and turning them, you would make it into a structure that sort of looks like this: a helix inside a helix inside a helix, and then there are enzymes that keep that tightly coiled together, and other enzymes that when you can clip it and send it spiraling apart so you can replicate it. And then the gene inside is our basic unit. We call it the basic unit of heredity. It's what codes for RNA, proteins, and enzymes. Everything that your body makes is basically coded for inside the DNA. And it's maybe a surprise that only about maybe 2 to 3% of the total DNA sequence are genes. And until recently, we didn't really know what the rest of it was. I mean, it was so-called junk DNA, but we're beginning to realize that there's really no such thing as junk DNA, that these sequences have a purpose, and we're getting to understand what the purpose of a lot of these non-gene sequences are. Okay. Let's talk about nucleotides. So this is the very smallest unit of a nucleic acid. And sort of like when we were talking about monosaccharides, with the very smallest unit of sugars and starches, these are the very smallest units of DNA. Now, you can see that they have sugars. And we know all about sugars now. So here's the basic sugar that is found in both RNA and DNA. It's called ribose. So it has 5 carbon atoms rather than the 6 we've been dealing with. So here's the anomeric carbon. We know it's the anomeric carbon because it is bound to 2 oxygens. And then carbon 2, carbon 3, carbon 4, and carbon 5. And so what we'll see is that carbon 1 will be attached to the base. And that is a base with rings that contain nitrogen atoms. And it's these rings that are involved in the hydrogen bonding that keeps our nucleotides together. It holds the 2 strands of DNA together. So we know about hydrogen bonding, and we'll see that hydrogen bonding is extremely important for how DNA is held together, how it's replicated, and the effect of mutations are found in the hydrogen bonding. So this is where the first carbon is where the nitrogen base will go. And then the 5' carbon is where the phosphate groups will go. And the phosphate groups will be held together by ester bonds. And we know that ester bonds have carbon, a double bonded oxygen, and then an oxygen coming off. Well, a phosphoester bond replaces the carbon with phosphate. And so it looks just like a carbon ester, except that it has a phosphate in the middle, except rather than a carbon. And so we call these phosphodiester bonds because there's a phosphate on, there's a phosphorus on either side and makes two of these bonds at the same time. Now, the most important difference between these two, between the sugars here, is to be found at the position of the 2' position. Ribose has a hydroxyl there. Deoxyribose does not. It has a hydrogen there instead. And that is the only difference between RNA and DNA, except for one other thing that I'll point out a little bit later. That is the difference, just that one oxygen atom. Everything else is pretty much identical. And so you'll see that this, because of, we know that this hydroxyl group can target a lot of other reactions to take place there. And so in RNA, this is much less stable because this hydroxyl group allows a lot of chemical reactions to take place there. A lot of enzymes bind there and use that hydroxyl group to catalyze reactions. And on DNA, there's a hydrogen there and they can't. And so DNA is much more stable than RNA. And that's one of the reasons why in us and pretty much every other living thing on Earth, we use DNA as the store of genetic information. And one of the reasons is it's a lot more chemically stable than RNA is. So these are the bases. Now you see they all have nitrogen in them, and they all have different ways of making hydrogen bonds. So, and there's two basic forms. There's pyrimidines, which have a single 6-member ring. And so these are the pyrimidines up here. So they have a single 6-member ring that contain nitrogen. Uracil, thymine, cytosine. And this is the other difference between RNA and DNA. It's the only other difference. Uracil is found, this particular molecule is only found in RNA. And the only difference between uracil and thymine is that methyl group right there. And that's more important than you can imagine. Because thymine in DNA itself is only supposed to be found inside the nucleus. And in fact, if the body detects the presence of, or the cell detects the presence of thymine outside of the nucleus, it commits suicide, which sounds a little harsh, but it starts a process called apoptosis. We're not really going to talk about that, but you may come across this a little bit later. Apoptosis is basically cell-mediated self-destruction. It sets off, like, you know, like you've seen, like how many bad movies where like the self-destruct button gets hit. That's basically the cell's self-destruct button. It's called apoptosis. If the cell detects something outside of where it normally should be, it senses that there's something is wrong with the cell, something is not right, and basically sends out the instruction to, you know, blow itself up. And so uracil is found outside the nucleus because you'd have to send RNA out of the nucleus in order to make protein out of it. This should not leave the nucleus, and the cell can tell the difference just by that single methyl group. So that's the first set of um, of bases. The second set are called purines. Purines have 2 rings rather than 1, still with the nitrogens in them. And there's 2 basic purines. One is called adenine and the other is guanine. You may notice that you've probably recognized these particular letters, A and G, C and T. And we'll see that A's, adenines, match up with thymine. They form linkage together through hydrogen bonds. And then C's interact with G's. And they form 3 hydrogen bonds between them. A's and T's form 2 hydrogen bonds. C's and G's form 3 hydrogen bonds between them. And so the fact that we know, like from our previous work, that hydrogen bonds are pretty strong. And so it's the number of these hydrogen bonds and their relative positions that make the DNA structure possible and allow us to make copies of the DNA. Because when we're making a new copy of DNA, we have to match the nucleotide sequence. And the nucleotide sequence is actually determined by its hydrogen bonding pattern. And if you don't match the hydrogen bonding pattern, it doesn't fit right, and there's an error. And so and we'll learn about, there's enzymes that do this, and they can tell whether or not the new nucleotide they add is stable or whether it's unstable. And if it's unstable, it gets tossed away. And a new nucleotide is added that's more stable. And it's all based on the hydrogen bonding. So the purine bases are A and G in DNA. And the pyrimidine bases are C and T. In RNA, we don't have thymine in RNA. We have uracil. And remember, the only difference is that thymine has that methyl group on it. But it hydrogen bonds in exactly the same way, in exactly the same space that thymine does, because we know that methyl groups can't hydrogen bond, and they don't interfere with the hydrogen bonding either. So everything is the same between them, DNA and RNA, except for the deoxyribose in DNA and the presence of thymine instead of uracil. Everything else is exactly the same. So these are what the nucleotides look like. And so this is where we join together the sugar part. So here's the sugar. There's so which sugar is that? Is that the ribose or deoxyribose?
Deoxyribose.
Why do you say that? How can you tell the difference?
It has that the OH number 2.
Mm-hmm. So if it's deoxyribose, what does deoxy mean? Deoxy means it's like missing an oxy, right? So if we look closer at the 2' carbon, you see it's got a hydroxyl group on it with an oxygen. So is that oxy or deoxy?
Sorry, that's oxy.
That's oxy, right. So that would be ribose. So these are all ribose sugars. And so you can see where on the sugar, we attach the phosphates and where we attach the base. So on the first carbon number 1, we attach the nitrogenous base. And on carbon number 5, we attach the phosphate. So this just has a single phosphate. So we call it, this is cytidine. And it just has a single phosphate on it. So we call it cytidine monophosphate, or CMP. The more phosphates we add, we then change the name. So if cytidine had another phosphate there, we call it diphosphate. And then if it had 3, we would call it triphosphate, or CTP. Now, the purines, adenine has a special place. You probably have heard of ATP before, I hope. Are people familiar with the ATP? They heard what it's for?
It's energy.
Yeah, it definitely is. It's basically the universal carrier of energy in the cell. It's not the highest energetic molecule. It's not the lowest energetic molecule. It's right in the middle. So it's able to transfer energy from high energy situations to lower energy situations. And it's just this molecule right here with another 2 more phosphates attached to it. And it's the addition and subtraction of these 2 phosphate groups that's basically responsible for all, practically all energy in your cell. It's when you get the phosphates added to you, you get energy, and then when you give those phosphates to somebody else, you lose energy. And for some reason, I'm not sure why it is, it's only adenine triphosphate that does that. GTP is also used a lot in the cell for signaling. Different molecules will signal each other with the presence of a GTP, but generally it's not used for energy. It's used for as a molecular switch. When a protein is binding GTP, it's on. When it's binding GMP, it's off. But it's not really used for energy. ATP, on the other hand, is basically all about the energy. But it's also used inside the nucleus to make DNA. So it's odd that it has both of those uses inside the cell. So here's our structure again, and you can see the difference between AMP, which is a single phosphate. ADP now has 2, and ATP now has 3. And these are what we call the phosphodiester bonds. And why are they called that? Well, because they have phosphorus in the middle. And you can see that it has an oxygen on both sides. So this phosphorus is making an ester bond there, and it's also making an ester bond there. So it's making an ester bond on both sides of the phosphorus. So we have a double bonded oxygen, just like we would have a double bonded carbon in an ester. And we have a single bonded oxygen on one side, a single bonded oxygen on the other side, and they're both linked to something else. So we call that a phosphodiester bond. And there's a lot of energy in that bond because we have a lot of oxygens there, all with pulling electrons away from the phosphorus. And we have a lot of negative charges. We have a negative charge there, one there, and one there. And so with all those negative charges, these groups are also pushing on each other. And there's a lot of strain. And when you relieve that strain by cutting these bonds, you release a lot of energy because you get to a much more stable place. Now, again, I just wanted to just reiterate the difference between ribose and deoxyribose, and just mention that it's this hydroxyl that allows, you remember what happens to the hydroxyl group in a carbohydrate. So if I have this bond and it gets oxidized, what does this form after it gets oxidized? you remember?
Aldehyde.
It forms either an aldehyde or a ketone, right? We form a carbonyl group. So now it's bonding even more to the oxygen. And if it's in the middle, this isn't on the end, so this would form a ketone. And then after a ketone is formed and it's oxidized even more, then what would it become? So if it gets oxidized even more, it gets bonded to another oxygen atom. So what do we call a carbonyl group with another oxygen attached to it? That would be this. What's this group called?
Carboxylic acid.
Carboxylic acid, right. And the only way you can form a carboxylic acid, the only way that carbon can form another bond is if it breaks this one. And so two cycles of oxidation on that carbon that has the hydroxyl goes from a hydroxyl group to a ketone to a carboxylic acid. And once it goes to the carboxylic acid, this carbon-carbon bond has to break. And then once you've broken that ring on the sugar, then the whole thing breaks down. And so that's why that doesn't happen on deoxyribose. We'll see that with how the nucleotides are put together, they use the 5' carbon, which is attached to the phosphate, and the 3' hydroxyl in order to extend and make long chains. That leaves this as the only carbon that isn't bound to something. And it doesn't have a hydroxyl group on it. And so it basically makes this completely resistant to oxidation at that site. Ribose, on the other hand, is open for oxidation on that site. Deoxyribose isn't. So we see the difference in stability there. So let's look at the structure. I think the structure really explains a lot of how these things work. And so nucleic acid is just a long polymer. It can be really, really long. Like in chromosomes, it can be, millions of nucleotides long. So DNA or deoxyribonucleic acid, now you know why it's called DNA. That's where the genetic information is stored. And then the RNA takes that information, copies it, transcribes it, and then it's responsible for transcribing or reading and then translating that genetic information into proteins and other things and other RNAs. So the DNA basically stays where it is. The DNA doesn't go anywhere. It stays in the nucleus. But the RNA gets made in the nucleus and then transported out where it does its job. So this is how we put these things together. So as I showed, nucleotides are linked together through these phosphodiester bonds. We have an ester on one side, we have an ester on the other side. So this would be a sequence. Notice that it's going between the 5', this is the 5' carbon, and this is the 3' carbon. And that's how we read DNA sequences, from the 5' end to the 3' end. That's the sequence of letters. We read them in that particular direction. So we would read this particular sequence as G, T, A, C, going from the 5' direction to the 3' direction. So you can see that since this is DNA, we just have a hydrogen there. And notice all the other carbons are bound up with something. So carbon 1 has the base on it. Carbon 2 doesn't have anything. Carbon 3 is where a new phosphodiester bond is made. Carbon 4 doesn't have a hydroxyl group on it. And carbon 5 is where the new, where the phosphate groups are kept. So everybody's doing something except for carbon 2. And so it's important that carbon 2 not be a target for attack and break the ring, the sugar ring open. So we're going to talk about the secondary structure of DNA. Anyone tell me what the primary structure of DNA is? What do we mean by primary, secondary structure of DNA? So if the secondary structure is this double helix structure, what's the primary structure? Have a guess at least.
It's 5' to 3'.
It's what?
It's just 5' to 3', like one strand.
It's just the sequence. Yeah, it's just the sequence. The sequence is the primary structure. And then the secondary structure is actually what structure does that take up now that we have 2 strands of DNA, because DNA is pretty much always double-stranded. That's another sign of something's going bad. If the cell detects single-stranded DNA in the cytoplasm, it also puts a gun to its head and shoots itself. So doesn't want thymine outside the nucleus, and it doesn't want single-stranded DNA outside the nucleus either. So yeah, so the primary structure is just the sequence. The secondary structure is the double helix. The tertiary structure would be how the double helix then is wound into this superstructure with proteins and everything else. So everybody was trying to figure out the structure of DNA in the late 40s, early 50s. It was sort of like the holy grail of biology, because like I sort of mentioned at the beginning of the chat today, we knew that genetics had been being studied since like the turn of the century. People were studying fruit fly genetics in the 1910s, and you probably have heard of the bean experiments, like back even before in the 1800s, where we sort of started to understand how genes worked. So we knew about genes, just didn't know what the hell they were. And then finally, DNA was found to be responsible for genetic information, but didn't know how it worked. And so one of the early experiments was done by a guy named Erwin Chargaff. It's an interesting fellow, not a very nice guy, kind of a difficult person to deal with. And he found out early on, I think probably like in the early 40s, that when he looked at the amount of adenine, he measured the amount of adenine and thymine and guanine and cytosine inside the cell. And he noticed that the amount of adenine was always the same as the amount of thymine, and the amount of guanine was always the same as the amount of cytosine. Now, they didn't match each other, like the amount of guanine wasn't the same as the amount of adenine, but if you add it together, A and T was always the same, G and C was always the same. So it seemed to indicate that there must be some interaction between A and T potentially, and G and C, but nobody really knew what it was. And so the number of purines always equals the number of pyrimidines. So T and C are your pyrimidines, A and G are your purines. And so A plus G is always going to equal C plus T. So that was known too. So no matter what cell you looked at, no matter what organism you looked at, it was always that those relationships always stood up. And so that was difficult to understand. And then, come on. No doubt you've heard of Watson and Crick. Everyone's heard of Watson and Crick. They published a structure. There's actually a lot of different structures have been proposed before then, including one by Linus Pauling, who, even though he won 2 Nobel Prizes, one in chemistry and one in peace, his structure for DNA was just fantastically wrong and kind of embarrassing, really, because he had all the phosphates on the inside. And we know that phosphates are negatively charged, and if you put them together in the middle, they're going to repel each other. So it didn't make any sense, but just goes to show you that great minds don't always have great ideas. So Watson and Crick came up with this model where the phosphates would be on the outside. So these are the phosphate groups. Oops. So these are the phosphate groups on the outside of the helix. And then the bases would be on the inside. And that way, G and C would be close together and could make their 3 hydrogen bonds. And A and T would be close together and make their 2 hydrogen bonds. And all of a sudden it became, oh, that's how it can replicate itself. Because if you pull these 2 strands apart, then they each have the information to create another strand. So all you need to do is pull them apart and then use that sequence to make yourself another copy. And that made perfect sense. What was really, really important about their discovery, is really kind of a tragic story, is that they stole their information on how the double helix was put together from a woman named Rosalind Franklin, which you maybe you've heard of, hopefully you've heard of. She did something called X-ray crystallography, which means she grew crystals of DNA and then shot X-rays at it and looked at the pattern that the X-rays made when they bounced off of the crystals of DNA. And she noticed that there was a regular pattern of X-ray coming off, and didn't know quite what to make of it. She knew that there was some sort of regular pattern, and her boss that she was working for at the time shared that data with Watson and Crick, who figured out that that regular pattern must be a helix, and then using models, basically cardboard cutout models of the different bases, they figured out that it must be a double helix. And so, and then poor Rosalind, she died before the Nobel Prize could be given out. The Nobel Prize was given, I think, about 7 years after the discovery. So Watson won a Nobel Prize, Crick won a Nobel Prize, Franklin's boss won a Nobel Prize, but she didn't, because the Nobel Committee does not give Nobel Prizes to dead people. And so, yeah, really a tragic thing. And one of the reasons why she died, because she worked with X-rays before people really knew how to protect themselves from X-rays very well. We know that like getting lots of X-rays is not very good for you. And so people weren't using enough shielding for doing X-ray studies back then, and she got cancer and died before she get the Nobel Prize. So so for me, it's like one of the saddest stories in chemistry. Sorry, I had to bum everybody out. So this structure, like I mentioned, explained really, really well how the different bases went together, because when they made their model, they could see that and like I said, they actually did this. They made a double helix out of metal balls and rods. And then in the middle, they put cardboard cutouts of thymine, adenine, cytosine. And they showed that the distances the bases would be from each other perfectly matched, not only the distance, but the relative orientation perfectly matched the maximum distance for hydrogen bonding. And so it's the most stable. Hydrogen bonds aren't stable beyond a certain distance. And then they're not stable if you get too close either, because then the atoms get too close to each other and repel. But the distance and orientation of the bases is exactly what you would want them to be to form hydrogen bonds. And so there's 2 hydrogen bonds between thymine and adenine, and you can see them here. So that carbonyl group is the hydrogen bond acceptor, and this amine is the hydrogen bond donor. And then here we have another hydrogen in between 2 nitrogens. So we have an amine here and a double bonded nitrogen there. And so it's the perfect distance. So cytosine and guanine are the perfect distance to make 3 hydrogen bonds. So 1, 2, 3. And so that explains how if you pull apart the 2 chains, just leaving the sequence, then this is how the new sequence could be formed, because wherever there's an adenine, a thymine would go next to it. Wherever there was a cytosine, a guanine would go next to it, and the other way around. And so we call those complementary bases. And they're complementary to each other in terms of the hydrogen bonds they make with each other. 2 for A and T, 3 for C and G. And that also explains in places where you want to melt the DNA, places where you want to pull the DNA apart a little bit so you can get proteins in there and start replicating, are rich in A and T. Now, why would proteins that are DNA sequence that is high in A and T melt at a lower temperature than a sequence that is high in G and C? Why would that be? So if I
Can you
have,
repeat that?
oh, go ahead.
Can you repeat that, Professor?
Oh, so if I have a sequence rich in A's and T's, that it's going to melt at a lower temperature, be easier to pull apart than a sequence that is rich in G's and C's. Why?
Is it because of the hydrogen bonds?
Elaborate. Yes, you're right. Why? So why? What's the
Thymine
difference?
Thymine and adenine has 2 hydrogen bonds compared to cytosine and guanine, which has 3.
Exactly. So more hydrogen bonds means more energy you need to put in order to break them. And so this is true wherever DNA needs to be pulled apart in order to either make another strand or to copy it into RNA. You will see this sequence that is rich in A's and T's. In fact, the protein I worked on in graduate school is the first protein to bind to start transcription. And I'll talk about that in a minute. But there is a sequence called the ta-ta box. And why is it called that? Because it has this particular sequence, pretty much every gene. And that's where a protein binds to it pulls those two apart and allows proteins to bind and start copying. And so if you change the sequence and put C's or G's in there, it basically shuts everything down. And so not only are the hydrogen bonds important for recognizing, for the bases to recognize each other, it's also important for maintaining the DNA. If you want to copy it, it's important if sequences you can pull apart a little easier. And if you want to make RNA copies, it's important to have an A and T rich sequence so you can pull them apart. All right, so let's look at this example. So as I mentioned, we always write sequences starting at the 5' end and then going toward the 3' end. So if I have a sequence here, so this would be a single-stranded DNA. We would just write that like this. If I want to make a double-stranded DNA, what would be the sequence that would go right underneath it here?
Three prime T T T T T T G G 5'.
Well, you're right about the sequence, but you're wrong about the 3' and 5', because now this is the 5' end, and that's the 3' end. Because
thought I
on
said
one
that.
strand, we're going in this direction, and on the other strand, we're going in that direction. But as far as the sequence goes, you're absolutely right. Because when we make a copy, when we make a copy of this strand up here, when we make a copy of this, we are going to make it, we always make the DNA and read the DNA in the 5' to 3' direction. So when we're making the copy, we go in this direction.
I think
We
I
go in
said it
this
right.
direction, and we start with the 5' and we go to the 3'. Okay, does that make sense? We always go 5' to 3'. The enzymes that make DNA and RNA always go in the 5' to 3' direction. That's why we always read the instructions, we read the sequence in that order as well. That's the only way they can make it, going from 5' to 3'.
Wait, so it's correct as far as like the transcription, but we all, because the 5 is on the end, we start naming it as 5' G G T T, etc. Is that what you're saying?
Well, no, it's just saying that if you want to recognize which ends are the 5' and the 3', if the top strand is 5' to 3 going in this direction, the bottom strand will be 5' to 3' going in the opposite direction. Have I lost everyone? Because that's how you make the copy. Enzymes that make DNA always go 5' to 3'. So if I have a sequence that goes from 5' to 3', it's going to start at the 3' end and go in the other direction, starting with its 5' and then going to the 3'. But that's the only direction it can go, is 5' to 3'. So if you're going to make a copy, it has to go in the opposite direction, 5' to 3'. That's the direction that enzymes work, making nucleic acids. Let's do the bottom one. So what would be the sequence for the bottom? Oh, Vera's got it. So yeah, C, 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 T, A. That is the 5' end of that sequence, and that is the 3' end of that sequence. Because that is the direction that all what we're going to learn are called polymerases. They're called polymerases because they make polymers. And so when they're making these polymers, they always go in the 5' to 3' direction. That's how they start with the 5' end, and they keep adding to the 3' end. So the 5' end is always going to have the phosphate on it. The 3' end is always going to have the free hydroxyl on it. So on the top, phosphate on that side, hydroxyl on that side. So you can always add to the hydroxyl side by adding another phosphate. You can't add to the phosphate side because the enzyme doesn't go in that direction. Okay, so if we were going to continue making this, we would go in that direction. And if we were going to make the bottom strand, we go in the opposite direction. All right, we'll deal a little bit more with that in a minute. So let's talk about replication and transcription. These are the basic two things that DNA is used for. So replication is just copying the DNA. So you're going to need it every time your cells divide, they need to make new chromosomes, right? So how do they do that? So we'll have to show you how that works. They basically pull the sequence, the double helix apart, and then build new sequences on both strands at the same time. And then transcription is just the process where you take RNA, you transcribe it in, or take DNA, transcribe it into RNA, and then that RNA is removed from the cell and then used for something else, either for making proteins or just for being RNA. We'll look at the different uses for RNA, the different structures. So here's a diagram for DNA replication. So you can see that we're pulling, so here we've got our double helix, and we're pulling it apart. We're basically unzipping it. And there's enzymes that do that called helicases. What do helicases do? They break up helixes. And so they basically go right here and help pull the DNA apart. And then we have enzymes called polymerases. Here we have a polymerase there, and we have some polymerases there. And notice what they're doing is they're binding to the single-stranded DNA, looking at the sequence, and then adding nucleotides that match, they're complementary to the sequence they're looking at. And they're doing it in the 5' to 3' direction. So wherever they see an A on the single-stranded DNA, they put a T. Where they see a G, they put a C. Where they see a C, they put a G. And they just continue going along. So from the one double helix, so here we have one double helix, we're making two. One here, the A helix, and another one here, the B helix. And so from each one of these new helices, A and B, has one old strand from helix one. One would be the 3' to 5'. The other one would be the 5' to 3'. Each one has one of those old strands and then a replica of the other strand. And we call that semi-conservative replication. And I'll sort of show you how that works. Actually, one of my professors from Oregon discovered this back in the late 50s, early 60s, very famous man. And I'll show you how it works. So here you have your original helix. Just imagine there's two strands there, okay? And so you pull that apart, and then you make two new helixes from it. So one would have one strand of that helix, and one would have the other strand of that helix. Now the new strands would be there. So each strand of the two new helixes, one is new and the other one is old. And so basically they were able to show that by taking DNA and then incubating it in the presence of nitrogen that was heavier. So normal nitrogen weighs 14, right? But if you grow these bugs in the presence of heavy nitrogen, they showed that the old strand had old nitrogen in it, and the new strands had heavy nitrogen in it, and proved that, because people didn't know whether one strand would be complete, one new helix would be completely the same as this one, and the other one would be a copy, or how it worked. And they showed that this is called semi-conservative replication. Each new helix has one strand of old and one strand of new. And it was the double helix structure that showed how that worked. So basically it goes back to the double helical structure, sort of explaining a lot. Once we saw that, it became readily apparent that that would be the way DNA would replicate itself. And then we needed to do more experiments to prove it. So that's replication, where you're just, all you're doing is making a copy of the DNA sequence that was there before. So you're forming new cells, you're going through cell division. You want to have the same DNA in your new cell as there was in the old cell, so you make a copy. Now, transcription is different. Transcription is you're taking DNA and you're making RNA out of it. So you're taking a DNA sequence and you're turning it into an RNA sequence. So RNA molecules are generally a lot shorter, at the most a few thousand bases, whereas DNA sequences can be millions and millions and millions of nucleotides long. Again, RNA is made from ribonucleotides with the 2' hydroxyl. And the newly synthesized RNA doesn't remain associated with the DNA, it leaves. So basically you pull the two strands apart, you copy only one of the strands. You don't copy both, you just make a RNA copy of one of them. And then it rezips up, it unmelts, it comes back together, and then that RNA leaves. And almost all RNA is single-stranded. You don't really see a lot of double-stranded RNA. You sometimes see double-stranded RNA in viruses where their genomes are RNA, but usually not. RNA is almost always single-stranded. And it doesn't remain in the nucleus, it leaves. So this basically gives you sort of a little idea of how the transcription works. So unlike replication, you've only got one polymerase this time, and it's only making a copy of one strand. So the DNA sequence that's used to make that the RNA polymerase recognizes and makes a copy of, we call that the template strand. So if the template strand goes CCTTA, the RNA that would be made from it would be GGAAT. So that would be the RNA strand going 5' to 3', and this would be the template strand going 5' to 3'. So then this RNA sequence would then leave, go outside the nucleus, and then do what it's going to do. So you have to unwind that sequence somewhere in the middle. And again, there's enzymes that do that, that help pull the two strands apart. And as I mentioned, there's normally an AT rich region that makes it easier to pull them apart. So, oh, sorry, there wouldn't be a T there. I made a mistake that nobody caught until just now. I wouldn't have a T in the RNA. What would I have instead?
U.
U, that's right. I would have a U there. So, because if there's a T there, that's not recognized as RNA, that would be a problem. So it would have a U. So uracil takes the place of T in RNA. And so there's a big complex enzyme called RNA polymerase that is a huge complex of like at least 20 to 30 different proteins that all come together and form this massive complex that's necessary to, you know, pull this apart and then do all these reactions. So there's three basic types of RNA. One you're probably the most familiar with, messenger RNA. I keep doing that. So you're probably familiar with messenger RNA. So that is the transcription of DNA that then goes into coding for proteins. So those are the RNA sequences that get translated into proteins. But that is not, notice that in E. coli is just the bacteria that scientists have been using for genetic studies for about 150 years. Only about 3% of the total RNA in E. coli is messenger RNA. Why is that? Well, it really only exists for a pretty short time because it is regulated. It's regulated at in the cell. It's regulated at the point of being made. So you only make messenger RNA when you need it. And then it's regulated again by how long its life is. And so generally you don't want messenger RNA to be around for a really long time because when you need the RNA, you make it, it makes the protein you want, then you want it to go away because you don't want to make that protein anymore. And so it's being continuously degraded. It's constantly being chewed up because you don't want excess protein being made. The whole point of doing the transcription is to make what you need and then stop. And so you don't see a lot of messenger RNA around because it's being made, being chewed up, being made, being chewed up. But what is being made and not being chewed up is something called ribosomal RNA. Now, who's heard of ribosomes?
They're proteins.
What do they
Protein
do?
synthesis.
Yeah, they're basically the big machines that make proteins. Ribosomes are really massive complexes that it's made of both protein and RNA, where messenger RNA goes in and is then translated into protein. And so actually most of the mass of ribosomes is actually RNA. And almost all of the RNA that you find in the cell at any one time is ribosomal RNA. And it's normally pretty big. You get these fairly large structures in there. And then finally, transfer RNA. Transfer RNA are also involved in making proteins. Transfer RNA, or called tRNA, have amino acids attached to them. And they go into the ribosomes. And when the tRNA sequence matches up with the mRNA sequence, that's where the ribosome knows when to add that particular amino acid. So that's how that works. And I'll go into a little bit more detail on that in a minute. So why don't we just take a quick 5-minute break? I'm going to have a quick drink of water, and then we'll come back in a minute, okay? and finish up. Oh, let me go back. All right. Let's talk about transcription and then translation. So I have replication there, which is kind of stupid. I should have I should have transcription. So once we have our messenger RNA, what are we going to do with it? So there's three, like I said, there's three basic types of RNA, and they're all going to be used as part of making proteins. So the ribosomes where all the action happens. And a codon, you may have heard of codons. That's the set of 3 nucleotides on the messenger RNA that tells the ribosome which specific nucleotide to add next. So that was discovered back, like I said, back in the late 50s, early 60s. We finally broke the genetic code and figured out, once we learned like a very old, not a fairly impractical way of sequencing DNA. It was a really arduous method, believe me. It involved all kinds of like bizarre chemistry and reactions you had to do with all these like really nasty toxic substances. Now we can, sequence your entire genome for like $100 in a matter of days. So it wasn't that easy like 50 years ago or 60 years ago. So once we figured out how the sequence, we could see that each amino acid had a particular set of 3 nucleotide instructions in order to, so if a gene had a particular sequence, we could see that led to this particular amino acid, that particular amino acid, next particular amino acid, because you compare the sequence of the gene and the sequence of the protein and match them up. So if a codon is on the messenger RNA, the anticodon, which recognizes it and is complementary to it, is on the transfer RNA. So the transfer RNA would have the complementary codon. So if the codon said GGG, the anticodon would have CCC, and that would be then linked to the amino acid that the codon is calling for. So let's go sort of have a look and see how that works. So here's what the transfer RNA. So you can see that these are pretty short. They're only like maybe 100 nucleotides or even shorter than that. And so on the 5' end, you've got your phosphate. And on the 3' end is where the amino acid goes. So this particular amino acid is called phenylalanine, and you can see that it has a benzene ring at the end. So it's important for making hydrophobic interactions. So the 5' end, it has the phosphate. The 3' end, it has the amino acid. And then it folds into a 3-dimensional shape that looks like this. So that's the 3-dimensional shape. So you can see on one end of it, it has the 3' anticodon. And on the other end, it has the amino acid that codes that particular sequence is identified with. So we have the anticodon loop at the bottom, and then the amino acid on the top. Okay, so that's what, and so each amino acid has a very specific transfer RNA that recognizes it and has this particular anticodon. And it's put together by another enzyme that recognizes the amino acid and recognizes the transfer RNA, makes sure that they match each other, and then makes this linkage right here. So that linkage makes sure that they're covalently attached to each other. And then it's this linkage that will be broken. This linkage right here is the one that'll be broken when it gets to the ribosome. And notice, what linkage is that? What is that? What kind of bond is that?
The ester bond?
That's an ester bond. That's right. And so when you add water to an ester bond, you get carboxylic acid back. And so that's why it's called an amino acid, because you have an amine there and you have an acid there. And it's those two that are linked together. Those are the two groups that link together to form proteins. You form a linkage between an amine and a carboxylic acid. And what kind of linkage is that?
Amide.
An amide. Man, you're crushing it. Yeah, an amide. We call it a peptide bond, but actually it's an amide bond. So see, you know all this stuff already. Just trying to put it together. So let's look and see how these proteins are made. So if you look at the sequence of a gene, you can break it down into a code of letters that codes for a protein. And we know that there's three nucleotides in each particular code. And so when we translate that code, we translate the RNA sequence into a protein sequence. So the messenger RNA has the sequence that dictates what the sequence of amino acids is going to be. And of course, that sequence originally came from the gene on the chromosome in the nucleus. So we need to build each one of these transfer RNAs. So here's our amino acid. So this particular amino acid, I believe, well, I can't even think what that particular one is, but it gets linked, as we mentioned, to the 3' end, because the 3' end has the hydroxyl on it. We basically always add anything to the 3' end because it has the hydroxyl group there, and we can form esters with it. The other end will have the phosphate on it. And pretty much the only thing we add to the phosphate is more phosphate. Oh, and that's a phone call that I will turn off. Actually, I'll just let it go to, sorry about that. So the linkage of a particular transfer RNA and a particular amino acid is made by an enzyme with a really incredibly imaginative name as an amino acyl tRNA synthetase. Why is it called a synthetase? Well, because it synthesizes synthesizes the link between a particular amino acid and particular tRNA. So you have to have the right amino acid attached to the right tRNA, or this whole system breaks down. It doesn't work. And so there's a lot of different tRNA synthetases. If there's 20 amino acids, how many synthetases are there? There's 20. And actually, new research, basically at my old research place in La Jolla, scientists are now making new amino acid tRNA synthetases and making their own codons. And so instead of just the 20 amino acids, they're making proteins with 21, 22, 23 different amino acids just by making these synthetic enzymes, which recognize a new sequence and attach a new amino acid to them. That's, you know, fairly exciting stuff. So these are the different steps. We'll go through the different steps. So if we look at the ribosome, so the ribosome is holding the messenger RNA and it's reading it. It's reading it in a 3 to 5 prime direction, so it can add new tRNAs in a 5 prime to 3 prime direction. So 5 prime, 3 prime. So it's adding them on. Oh, sorry, other way around. 5 to 3. 5 to 3. So it looks for a particular sequence in the mRNA. The tRNA binds to the complementary sequence. So if I have GAU, it is going to bond to AUC. They recognize each other, and that codes for aspartic acid. So aspartic acid is one of the 20 amino acids. It goes there. The next one coming in recognizes AAA. So it would be UUU. UUU is the anticodon for lysine. So then lysine gets added. And you can see that each time we add a new amino acid, we break this bond. The empty transfer RNA leaves, and we form a bond, a peptide bond, an amide bond there. And we just keep adding them on one after another, looking for the next set, the next triplet set of codons. So the next one would be UUC, and so that would correspond to GAA or phenylalanine. So that would come in next. So every time one comes in, forms, recognizes the triplet code, adds the amino acid, and then leaves out the other side. So when it forms that peptide bond, at the same time, it breaks the bond at the 3' end of the transfer RNA where the amino acid was. And so now it's empty and it gets shoved out the other side. And we just keep continuing the process. We just keep going. So I've already asked you this particular question. So you've already nailed that one. So this just sort of shows a little bit better how these amino acids form the amide bond here between the carboxylic part of the amino acid, the acid part of the amino acid, and the amide or the amine, NH2, and the amine part of the amino acid. And when you make that bond, you break at the same time, you break the bond of the amino acid have with the transfer RNA, it leaves, and you just get a longer and longer and longer chain. Now, when does it stop? Well, it stops when you finally come to a particular codon. And what it's looking for is a termination codon. Once you come to a termination codon, there's no transfer RNA that recognizes that particular sequence. And so when the termination codon comes up, or a stop codon comes up, there's no tRNA that recognizes it. And so then you're done. And it's released. So that would be like the last tRNA would have added its amino acid. You get to a stop codon that releases the last tRNA, and then you're finished. So here we actually have something called a release factor, then recognizes the stop codon. And when it recognizes the stop codon, it breaks apart a little part of the ribosome, the small subunit of the ribosome, and it releases the messenger RNA. And when the messenger RNA gets released, it gets chewed up because it's done its function. It's finished. It's made the protein that it was supposed to make. And so if you need to do this again, you don't use the same RNA, you use a new one. So if you need to make more protein, you make more RNA. You generally don't use the same RNA over and over. It gets copied, it gets translated once and then destroyed after that. So here's the code that was worked out many, many years ago. So there's 61 codons that have been determined so far. Notice that 3 of them cause termination. So there is these 2 calls for stop and this one. So UAA is a stop codon and UAG is a stop codon and UGG is a stop codon. So anytime you see one of those, you know your protein is done. That's going to stop. Interestingly, only 2 amino acids have just a single codon. One is called methionine and the other is tryptophan. Interestingly, every single protein starts with this codon. That's also called a start codon. Every single protein starts with a methionine. So basically, that particular codon tells that's the starting sequence for the ribosome. Once it sees that sequence, it says, this is where I'm going to start. And so methionine is always the very, very first amino acid to be incorporated. Every other amino acid has 2 or more. Some of them have like 4. Why that is, we really have no idea. But the interesting thing is, and this is like, if you want evidence of evolution, every single living thing on Earth has the same genetic code. From, E. coli to us, everyone uses the same viruses, all use the same genetic code. So something's been passed down for a really, really long time. So let's look at how these sequences can change, how they can be mutated, and what happens when that takes place. So a mutation is basically any physical or chemical change that alters the nucleotide sequence. That's all it means. A mutation is just a change in the nucleotide sequence of DNA. But there's a lot of different kinds. You could have a point mutation where a single nucleotide is either substituted, like an A becomes a C or A becomes a T, or it gets deleted or it gets added. So all with a single nucleotide. And then you could also have much larger mutation. You could also have like big pieces taken out. So it doesn't have to be just a single nucleotide. You can have a whole sequence be removed. Generally, most naturally occurring mutations are point mutations with a single nucleotide. And a mutagen or a teratogen is something that causes mutations. And so like benzene, for instance, is really, really bad for us because of that ring gets stuck in between the rings, the nucleotide rings. And when the DNA is being copied, it causes mistakes to get made because you have that extra ring in there between the nucleotide rings that's forcing them apart. And so you have errors being made. Other mutagens actually cause chemical damage directly to the nucleotide bases. They will add a methyl group or an ethyl group or something like that in a space where hydrogen bonds were supposed to be made. Now those hydrogen bonds can't be made anymore because of the presence of an ethyl methyl or whatever group there is. And so it screws up the replication. If you have only 2 hydrogen bonding sites where you used to have 3, then it's not going to be recognized the same way, and it'll put in the wrong base. And a lot of times these will cause genetic disease, either by substituting the wrong amino acid. Instead of like a glycine, like there'll be a histidine will be put in there, and it'll change the function of the protein. Or what happens even more commonly is that it inserts a stop codon in there. And so you only make half the protein rather than the whole thing. And if you don't make the whole thing, generally it just doesn't fold properly into 3-dimensional shape, and it just gets degraded. So then you wind up making no enzyme at all. So here we have substitutions, insertions, and deletions. So if this is our normal sequence up here, so we'll just use this as that'll be the template we'll look at. Now a substitution would change, let's say we change this C to a T. So instead of glutamic acid, we're now putting a lysine in there. So we've gone from GAA to, GAA to AAA on the mRNA. Now that's going to make a huge difference, because glutamic acid is an acidic. It's an acidic amino acid. It adds a carboxyl group somewhere in the protein. Lysine is a basic amino acid. It adds an amino group somewhere in the protein. So you go from having a negative charge to having a positive charge. And instead of having a carboxyl group, you've got an amine group. So that would have a dramatic change on the protein, especially if this is involved with actually doing an enzymatic reaction. A lot of times in what we call the active site of enzymes, they need side chains from these amino acids that can actually do some chemistry. And we know that this can do some chemistry, right? There's a lot of reactions that carboxylic acids can do. There's a lot of reactions that amines can do, but they're not the same ones. So that would have a dramatic effect. But let's look and see if we had an insertion instead. So instead of just switching this C to a T, let's just add another C. So if we add another one, now we've got CCT. And not only have we changed that one amino acid, we have changed every amino acid downstream from there. So now we've changed this glucine to a glycine. And since we've added another amino acid, we've screwed up the whole triplet codon all the way down the line, because we've added a new letter for that codon. Now we've added a new letter for this one, and the next one, and the next one, and the next one. And so substitution mutations can be bad, but generally they're not nearly as bad as insertions and deletions, because the substitution mutation will just change one amino acid. And it might not even change that one amino acid, because remember, there's some redundancy in the codes. Some amino acids have 2, 3 different codons. So you may just change the codon from tyrosine to another codon for tyrosine. No harm, no foul. So that wouldn't change anything. But an insertion or a deletion, that is going to move the entire chain. You're either going to add a new letter to every single codon from now on, or you're going to delete one from every single codon, but you're going to change everything. downstream. So you can see that insertions and deletions are much more deleterious, much worse than a simple substitution. And these are just a list. I mean, probably familiar with a whole bunch of different genetic diseases, some worse than others. Like hemophilia is caused by mutation that changes the clotting factors in your blood, either factor 8 or factor 9. And so the blood doesn't clot as quickly or at all. And so, yeah, these people are like, always in danger. If a cut can kill them because the bleeding won't stop. So they have to have these clotting factors added to their blood in order to get it to the clot. And then there's just, sickle cell is a single mutation in hemoglobin. And it changes the shape of the hemoglobin into a sickle cell shape. So you have the regular hemoglobin, and then the regular hemoglobin will sort of be like a flat disc, and then the sickle looks like that. And that's just a single amino acid change, basically bends the whole thing. And there's a whole bunch of different ones. And so now we're working on whether we do have the ability to change these sequences back now. And now the question is, should we do that or not? Because if we change the sequence, we can change the genetic sequences going forwards, and that's kind of a dangerous thing. We don't know if we should do that or not. You might want to be interested in finding more about that. So at the end, let's just wrap up with viruses. I find this really, really interesting. So viruses are fascinating in that they're not actually living things. The other things that make us sick, like bacteria and mold and fungi and all that kind of stuff, they grow inside you and make you sick. But viruses can't replicate themselves. They don't grow and replicate themselves by themselves. They need a host in order to do that. So basically, all a virus is just a coat of proteins with some genetic material inside, and that's it. It's not a cell. There's nothing like that. It's just genetic material with a protein core on the top. And so a retrovirus is a virus made of RNA. Most viruses are made of DNA for the same reason that we use DNA, because it's more stable. But the really, really simple viruses are called retroviruses that basically have short, single-stranded, in some instances double-stranded RNA, what they call genome. So all of their genetic material is RNA. And so when it goes into a host cell, it infects it with RNA, and that RNA then is turned back into DNA, replicated, and then made into RNA again to make more virus. We'll sort of go into how that works. So these are really, 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 really small, much, much smaller than bacteria for the most part. There are some new viruses been discovered in like the last couple of years that are massive. Interestingly, these supermassive viruses only infect these supermassive single cell organisms. It's all very weird. And only been found like about the last 5 years or so. It's really, really interesting. So they have either, like I said, either have DNA, most of them have DNA, or they have RNA, but they never have both. You never see both. They only have one or the other. And that shows that they're using the RNA in there as their genetic material. So DNA and RNA viruses. Like I said, we also call RNA viruses retroviruses. We call them retroviruses because they need to be made back into DNA before they can be replicated. And there's a very special enzyme that does that I will mention in a sec. So this is your typical life cycle. So you've got your virus, and it's got its protein on the outside and its genetic material on the inside. Now it recognizes something on the host cell. Now that is like something that's always of interest to virologists, because they want to recognize, there's usually some very specific protein on the surface of the host cell that the virus recognizes, binds to it, and then the virus gets pulled inside the cell. So usually we call it some sort of receptor on the series on the host cell the virus recognizes, and that's been identified with COVID-19. So the virus now injects its material inside the cell. It doesn't even have to enter the cell. Sometimes it just injects its RNA. Now, when the RNA is in there, now we have single-stranded RNA that needs to be copied into DNA. So then it can be transcribed back into RNA. Because we don't have an enzyme that takes single-stranded RNA and makes a copy of it. And that wouldn't be of any use to the virus anyway, because it needs this exact same sequence. So if we were to make a copy of that, it would be of a complementary strand, which is not what the virus wants. So in order for the virus to replicate, it needs that exact sequence. So it gets turned into DNA, and then that DNA gets turned into an exact copy of the, that DNA gets copied into the RNA. And that requires an enzyme that we don't have in our body. It's called reverse transcriptase. And what that does is it takes RNA and turns it into DNA. Now that's something we don't do because there's no reason for us to. We take RNA and turn it into, we take DNA and turn it into RNA, then the RNA leaves and then gets degraded. We don't really have a reason to take RNA and turn it back into DNA, but retroviruses do. They need that in order to replicate themselves. And so they actually carry the gene for that with them. That is part of their genetic material in what's called the Pol gene, Pol for polymerase. And so basically it encodes for the enzyme that it will use to replicate itself. And it needs it because we don't have an enzyme like that. And so this is also a very, very important target for particular viruses. Since we don't have that enzyme and viruses do, if we can figure out a way to shut this down, then it will shut down replication of the virus, and it shouldn't hurt us in particular. So that reverse engineered or reverse transcribed DNA then goes into our genome. It gets integrated into our genome. And then we make RNA out of it. And then that RNA makes more polymerase, it makes more single-stranded RNA. And then when there's enough single-stranded RNA, then we also make the proteins on the outside. We make that for them. It packages itself, and then it basically blows up the cell on its way out. So it'll make like several thousand or million copies of itself. And once it's made enough copies, basically just bursts the cell and then goes somewhere else, either to infect another cell in you or to get coughed out and infect somebody else. But it's the reverse transcriptase I really want to talk about because it's the most interesting part. And so until recently, probably the best known retrovirus was human immunodeficiency virus. And it was obviously, studied to death because it was like, it's still, killing, hundreds of thousands of people around the world. But that virus uses proteins on its outer surface to bind a surface of T cells. Now, T cells are a group of white blood cells that normally, this is the really evil part of HIV, it basically infects the exact cell that would normally be used to destroy it. And so normally what T cells do is that they recognize cells that have something wrong with them, cells that have something on their outer surface that isn't quite right. And so when HIV infects these T cells, then they can't recognize HIV anymore because it's actually infected the cell that's supposed to take it out. So then it replicates, it destroys the T cells. That's normally what kills the people that have it, so that their T cell count drops to basically 0, and then they get infected by something that normally isn't particularly harmful. That was the real thing that made scientists and other physicians not understand what was going on. They had all these people who were dying of these like illnesses that no one had, that people normally get and recover from easily, and it hadn't killed anyone in like 50 to 100 years. These really, really rare diseases, until it was discovered that it was because the T cells that normally take out these regular things that are perfectly harmless to the rest of us because we have immunity against, their immunity was completely destroyed. And so like you see a lot of people dying from pneumonia, other sort of normal infectious disease like a flu would just basically be fatal to people. So what came along to sort of help with that was a target on the reverse transcriptase of HIV. So just like every other retrovirus, it has a Pol gene for reverse transcriptase. And so if you had something that would bind to reverse transcriptase and prevent its RNA being made into DNA, that would be a good way to stop replication of the virus. And so the very first drug, you're probably familiar, especially growing up in the Bay Area, AZT was the very first thing that was made, and it looks like TTP, but it stops, instead of having it doesn't have a 3' hydroxyl. So we know that everything goes 5' to 3', and we add things to the 3' hydroxyl group. So if you can't add anything, then it stops. So it stops replication. Unfortunately, it's not a silver bullet, because it also can work its way into the nucleus and sort of screw up replication there as well, though it has a much higher affinity for reverse transcriptase than it does for regular DNA polymerase. So that works. That was the very first drug that worked. Now we've got a whole bunch of other ones that work even better, that fit reverse transcriptase even better than that one. Now, unfortunately, we haven't been able to find a way to completely eliminate HIV. So we have these drug cocktails that inhibit reverse transcriptase, but people need to take them basically for the rest of their lives. And the reason for that is, since they destroy T cells, it's really difficult to make a vaccine that T cells will recognize. So people are still working on it, and I hear there's new human trials going on all the time, but hopefully like the next 5 to 10 years we'll have one. Now that brings me to COVID. Now that's been a problem for us recently with the new Omicron strain. Now, where do these come from? You probably are wondering yourself, right? We had the Delta strain, now we've got the Omicron strain. What the hell do all these mean? Now, this is a coronavirus, and they get the name because when you look at them under an electron microscope, it looks like they have this sort of crown or halo around them. And that is just the spikes on the surface of the virus coming out, kind of makes it look like looks like a crown. Now, it targets the alveoli cells, or really, the small air sac cells in the lungs. Appears to be the target for COVID-19. Now, why do you get so many more new strains of this particular virus when you don't really see that with other viruses? You kind of do with the flu as well. Every year we sort of like get a new strain, but it seems like we get a new strain of this like every couple months. Well, why is that? And it comes down to reverse transcriptase. So here is what reverse transcriptase looks like. And it was in 1975 that we finally found an enzyme that converted RNA in retroviruses to DNA. And it was found in cells that were infected with retroviruses and caused tumors. We now know that some cancers are caused by retroviruses. So we know it's encoded in the Paul gene. And now what's unique about reverse transcriptase? Not only is it the only enzyme that takes RNA and turns it into DNA, it also doesn't have an editing function. DNA polymerases have an editing function. So not only can they go 5' to 3', they can also cut out 3' to 5'. So if they find an error in them, they can back up and take some of those sequences out and then remake them. If the H bonding isn't right, if they don't fit properly in the polymerase active site, it can go backwards and correct it. Reverse transcriptase can't do that. And so basically it just makes a copy and that's it. It doesn't go back and fix it. And so the reverse transcriptase in HIV has been found to have an error rate of 1 in every 2 to 4,000 bases. That doesn't sound like it's too bad, but just think about it. Every time the virus replicates, it makes an average of 5 mistakes. That's 5 mutations every time it makes one new version of itself. That's why COVID-19 is so prone to mutation, because reverse transcriptase makes a ton of mistakes when it's converting the RNA into DNA. And so it's accumulating mutations at an alarming rate. And in fact, this new Omicron strain appears to have about 50 mutations in it, in just the spike protein. And that's just remarkable. I mean, the first time I read that, I thought it had to be a mistake, but no, just the spike protein alone has 50 mutations in it. And so we don't know, at this point, we still don't know whether or not it's more, it's going to cause more severe disease. The very first indications from South Africa are that it causes less severe disease, but that was in a population of mostly healthy younger people. So we don't really know that yet. It appears that it spreads more easily, but we don't know whether it's because it replicates more quickly or something else. And we don't know how well it will be recognized by the antibodies we have from the shots we took. So maybe good news, it may be bad news. We just don't know enough yet.
I was just wondering if does it also have to do with the fact that the protein knocks the ACE2 receptor? Angiotensin, I think it was angiotensin.
Yeah, that's ACE.
And that's the reason why it has a higher binding affinity. If you compare it to like SARS. which its affinity isn't as high compared to COVID.
Yeah, that could be one reason why it replicates better. So, I mean, yeah, so obviously if you had mutations that changed made less binding, those wouldn't be selected for because then they wouldn't work as well. So basically we're seeing natural selection in action. So these things make so many errors that they're subject to natural selection very, very harshly. So any error that doesn't help it replicate better gets lost. Any error that helps it replicate better gets kept. Any error that helps it evade the immune system better gets kept. So yeah, that's one of the reasons for concern is that, yeah, you get so many mutations so quickly that it can evade immunity really, really quickly. But we don't know enough about this one yet.